So coming on to part 3, we'll look at the loading of the mRNA, miRNA and siRNA single strand into the RISC complex. So, sorry, we don't actually know that much about the RISC complex, but there are some components that we've identified that are important. So one of them is the Argonaut proteins. I should probably say R, grammatical accuracy. So the Argonaut or AGO proteins, which are the principal catalytic components of the RISC complex. It's responsible for cleaving the messenger RNA. Again, they belong to an evolutionarily conserved family, and it's defined by the presence of a Paz domain and a Peewee domain. And there are multiple homologs in each metazoan species, which may or may not, we don't actually know, but we suspect it could have tissue-specific or developmentally regulatory roles. So there have been 5 found in Drosophila melanogaster, 8 in mammals, and 24 in C. elegans. Also found to be an important component of the RISC complex are the RNA binding proteins VIG and Fragile X related protein. And then we also have the nuclease Tudor SN. And I guess temporarily the mature miRNA single strand also belongs to the complex, but only temporarily, obviously. So the mature miRNA strand from the duplex is selectively incorporated into the RISC complex for target recognition, and this ensures both specificity and probably functional efficiency. The miRNA asterisk strand, which was the passenger strand, is probably degraded rapidly upon its exclusion from RISC, as though, well, there have been very low recovery rates of it in endogenous tissue. As DICER processes the pre-mRNA into the duplex, the stability of the 5' ends of the 2 arms of the duplex is usually different. Whilst the miRNA, the mature miRNA can reside on either strand of the hairpin stem, it's almost always derived from the strand with a less stable 5' end compared to the other strand. In the rare cases where both strands have similar 5' end stability, then each arm is predicted to be assembled into RISC at similar frequencies. This thermodynamic model also applies to the asymmetric assembly of the siRNA duplex. So you've got one strand of siRNA that's less stable than the other and is preferentially assembled into the RISC complex to target mRNA cleavage. So really there seems to be a common mechanism here that regulates the asymmetric assembly from double-stranded RNA duplexes. Coming on to the final parts, the fourth and final, post-transcriptional repression by miRNAs. So in animals you've got miRNAs that repress translation of mRNA, messenger RNA, into protein, but it's not clear exactly at what stage this occurs. There's only one plant miRNA that does this as well, MIR172. Some lines of evidence suggest that the repression occurs after translation initiation, probably during the translational elongation and or the subsequent release of the protein. One mammalian exception, miRNA MIR196, cleaves the mRNA of HOXB8, its target gene, so it acts like siRNA. In plants, though, most mRNA destroy the target mRNA, so act like siRNA from animals, and have near perfect complementarity with sites located throughout the transcribed region of the target gene, instead of being limited to the 3' UTRs. So there you go. Once you know the gene you want, you get the sequence that will destroy the messenger RNA that produces that protein. So that's it for today.